Have you ever wondered how scientists detect genetic disorders like Huntington's disease? It all starts with a fascinating process, analyzing our DNA. Huntington's disease is a neurodegenerative condition caused by a mutation in a single gene. But how do we uncover this mutation? Through the power of molecular biology, using techniques like PCR and gel electrophoresis, scientists can pinpoint genetic abnormalities with incredible precision. In today's video, we'll break down how this testing works, showing how DNA analysis can identify carriers and provide insights into genetic disorders. Stick around, it's simpler than you think. Huntington's disease is caused by a mutation in the HTT gene, located on chromosome 4. This gene contains repeated sequences of CAG, which code for the amino acid glutamine. In a healthy individual, there are fewer than 26 of these repeats. However, in someone with Huntington's disease, these repeats expand to 40 or more, causing the production of a toxic version of the Huntington protein. This mutation leads to the progressive breakdown of nerve cells in the brain, particularly in the basal ganglia. Symptoms often start between the ages of 30 and 50, including involuntary movements, difficulty with coordination, and cognitive decline. The disease follows an autosomal dominant inheritance pattern, meaning a child has a 50% chance of inheriting it if one parent carries the mutated gene. To detect the genetic mutation associated with Huntington's disease, scientists use a technique called polymerase chain reaction, or PCR. This process allows them to amplify specific DNA segments, making millions of copies of a target sequence. Here's how it works. First, the DNA is extracted from a sample, such as blood or saliva. Short primers, designed to bind to the ends of the HTT gene's CAG repeat region, are added. The mixture is then heated to separate the DNA strands in a process called denaturation. After cooling, the primers attach to the single strands during the annealing step, and a heat-resistant enzyme called Taq polymerase synthesizes new DNA strands in the extension phase. Once amplified, the DNA is subjected to gel electrophoresis, a technique that separates DNA fragments by size. A gel made of agarose is placed in an electric field, and the DNA samples are loaded into small wells. Since DNA is negatively charged, the fragments move toward the positive electrode. Smaller fragments travel faster and farther than larger ones, creating distinct bands that reveal the number of CAG repeats. By comparing these bands to a control, scientists can determine whether someone carries the mutation for Huntington's disease. Genetic testing has revolutionized our understanding of diseases like Huntington's. By identifying mutations early, we can offer better care and empower individuals with knowledge about their genetic health. Whether you are a student or simply curious about science, exploring these techniques can open doors to understanding how our DNA shapes us. If you enjoyed learning about this process, don't forget to like, share, and subscribe for more fascinating insights into the world of genetics and molecular biology.